Hi everyone, my name is Anushka, and my creative project is on photosynthesis and focusing more on the light dependent reaction. Photosynthesis is a two stage process, the light dependent reaction and the Calvin cycle. The light dependent reaction, as the name implies, requires light. Energy from sunlight is absorbed by chlorophyll and stored into energy called NADPH, an electron carrying molecule, and ATP. The light-dependent reaction takes place in the thylakoid membrane, in the granum, within the chloroplast. A photon of light travels until it reaches a molecule of chlorophyll, and then a molecule of water is split due to the photon of light. This splitting releases an electron, resulting in the formation of oxygen and hydrogen ions in the thylakoid space. After this process, the two energy carrying molecules NADPH and ATP are utilized in the series of chemical reaction called Calvin cycle. In the Calvin cycle, the plants take carbon dioxide from air and make sugars such as glucose and sucrose. These sugars can be stored for later use by the plant as an energy source to fuel its metabolism and growth. In this experiment, we will primarily focus on light dependent reaction. For this experiment, I have chosen a spinach leaf to be the subject of this experiment because of its high concentration of chlorophyll and non-waxy cuticle. In the glass jar, I've added 1/3 cup of room temperature water along with 1/16 teaspoon of baking soda. Sodium bicarbonate is added to increase the amount of carbon dioxide in the water so that the light dependent reaction can take place. And then I am also adding a drop of dish soap. Dish soap is added to reduce the surface tension of the leaf, allowing water to move freely in and out of the cells. First, I'm going to remove all the air pockets from the spinach disc by creating a vacuum with the help of a syringe. In the syringe, I have water and bicarbonate solution with 10 spinach discs. And by pulling the plunger, I have essentially created a vacuum in which air in the spinach leaves are dispelled because of hypertonicity. As you can see, all of the discs have sunk to the bottom of the syringe, implying that most, if not all, of the air are expelled from the disc. Then we pour into a beaker. Since I don't have one, let's use a glass jar. Right now, all the discs sank because the air has been removed from their internal space. As photosynthesis occurs, oxygen is produced as a byproduct. This oxygen accumulates in the leaf discs, causing them to be buoyant and gradually rise to the surface. I performed this experiment 3 times, and every single time the first leaf started to float at 30 to 45 minutes. The times are significantly larger than the other experiments that I've seen. I believe that it may be due to the inadequate lighting and not having a beaker. If the beaker was if a beaker was used, then the thickness of the water layer may be much smaller in comparison to my glass jar.